explaining, clarifying a bit more about what we just talked about transcription, particularly strand directionality. So things you're going to need to be able to answer questions about, be able to identify the template, also called antisense strand, and the coding/sense strands from an image. Determine the direction of transcription versus how the RNA is being built. What is what we call the direction of transcription? What is that relative to versus the direction that RNA is growing? And what do upstream and downstream mean when we're talking? Okay, so some important points of clarification about directionality. When we're talking about transcription, we are thinking about DNA, which is double-stranded, and then RNA, which is single-stranded. We're thinking about 3' to 5' and 5' to 3'. So let's take a moment to make sure that we're really grounded and understand these concepts and the directionality. When we talk about the direction of transcription, which you can see right here, is this arrow. So I've made it kind of yellow. I don't know, that makes it sort of hard to see. I wanted to highlight it, but I just made it sort of invisible. The direction of transcription, you can see that this arrow is pointing to the right of your screen. What the heck does that mean? There are so many different things that we're thinking about. What is that arrow describing, or what is it relative to? Is something that it's so easy to get confused about and get lost in. So let's orient ourselves. Our first thing to do is orient ourselves when we're looking at the DNA. So it says over here on the left, it already gives you these answers. But stop and think for a second when you're looking at this image. If you didn't have that information over on the left, how would you be able to figure out which is the template strand and which is the coding strand? How would you know? The way you would know is that this is the strand that RNA polymerase is creating an RNA, a pre-mRNA from. So then that is the strand that is our template, also called the antisense strand. So remember from an earlier video, it's called an antisense strand because we can't directly read it and know what our RNA what our RNA will be. Then this other strand is the coding strand. /sense strand. And it's called this coding or sense strand because we can read it and know exactly what our RNA transcript is going to be. So now that we've oriented ourselves to what is the template strand and what is the coding strand, we can sort of start to think about the direction of transcription. The template or antisense strand is running 3' to 5'. Whereas the coding strand is running 5' to 3'. So the direction of transcription is relative to the template strand. So you can kind of think of this as the direction that you would read, because that's what RNA polymerase is doing, is it's reading the strand. So what direction is RNA reading? So direction of transcription question you can ask yourself is, which direction is RNA reading? I mean, not, did I say that? Did I say RNA? RNA polymerase. I apologize if I just said RNA. And an abbreviation, a way that we very, very commonly talk about polymerases is we'll just say RNA pol. That's short for polymerase. So which direction is RNA polymerase reading? Looking here, RNA polymerase, it's going to be reading the template or antisense strand. The template or antisense strand is running 3' to 5'. Therefore, the direction of transcription is 3' to 5'. So RNA polymerase, I'm going to use a purple, is moving this way along the template strand. It's reading from 3' to 5'. Sort of like when you read a book, you read from left to right. Okay, hopefully we're good there. So now we're going to think about what's happening with that pre-mRNA that RNA polymerase is producing. The whole point of transcription is to produce that RNA. Thinking about the pre-mRNA right here, remember that the RNA is anti-parallel. It's anti-parallel to the template strand. So if the RNA produced is anti-parallel to the template strand, that means its primes are going to be flipped. And you can see that here. This end is 5' and this end is 3'. The other way from the template strand. So RNA polymerase can only add nucleotides to the 3' end of the growing RNA, right? So it can only add, it's getting crowded, this image is getting crowded. It can only add here. So RNA polymerase, when we're thinking about the RNA polymerase is extending or growing that strand on the 3' end of the RNA, while it's moving 3' to 5' on the DNA. So think of it as sort of like clamped onto the DNA and ratcheting down. And the direction that it's ratcheting is toward the 5'. And it's producing an anti-parallel strand, so it's extending that RNA at the 3'. So when we talk about the direction of transcription, we're almost always talking about it relative to the template strand. The direction of transcription is 3' to 5' on the template strand. The mRNA is added to on the 3' end. So if this is our template strand, what is our, and let's say, I get all caught up trying to pick the right colors. No, I shouldn't use purple. We'll use gray. This little thing right here, this is where RNA polymerase is going to come bind. What's it called? The whole section, not the little part in it. What is this section called? So this section would be the promoter, right? So RNA polymerase is going to come bind. Here we go. Here's our RNA polymerase. comes and binds the promoter, and as you know from earlier videos, specifically the consensus sequence within the promoter. That's the specific part that it identifies. And then it's going to move this direction, okay? Cool. Then once it hits the beginning of the transcribed region, the beginning of the transcribed region is indicated by a what? How do we know where transcription actually starts? So the beginning of the transcribed region is labeled how? What do we use to designate that? It's indicated by a +1. And remember that anything upstream of that +1, or anything before that +1 is upstream, anything after is downstream. So it's indicated by a +1, and then from downstream from that +1, we have our transcribed region. It hits that start transcription start, that +1. As it's moving in the 5' direction, it's the first nucleotide that it lays down is 5' to what we would call the left of your screen. And then it keeps adding nucleotides because it can only add them to the 3' end. So it's moving in, oh, I shouldn't use that color. It's moving in this direction. It's adding bases to the mRNA, extending that mRNA, adding to the 3' end. So as it moves along the DNA that it's transcribing, the strand of DNA that it's transcribing, moving toward the 5' end, the growing mRNA is going to exit the RNA polymerase, which is what this little drawing is kind of showing, and it's going to keep adding to that 3' end, and it's going to keep doing that until it reaches that sequence that tells it needs to terminate, it needs to stop transcribing. So then looking at this strand of DNA, this template strand, we just talked about this, and we talked about this in an earlier video. What is everything toward the 3' of the +1? What is this called? This is called upstream. Oh, I can't spell. You guys have figured that out, right? You would think stream I could spell though. So everything toward the 3' end of the +1 is considered upstream. And so then what is everything to the 5' end all the way to the termination sequence? What is that called? It's called downstream.